Hi guys. I'll start the class in maybe like 2-3 minutes after a few more people have joined.
Using guide. अभी तो
Okay, I hope you guys can hear me. I'll get started with the class now. So we'll continue from where we left off, which was with the respiratory system. So last time we had talked about the anatomy of the respiratory system, the upper respiratory tract, and its different components, and then the lower respiratory tract. And so I won't spend too much time recapping because there's quite a lot to cover. Um, so we'll move on to where we had stopped last time. So we had stopped at talking about hemoglobin, right? So we know that oxygen is transported in the blood via two different ways, the majority of it being bound to hemoglobin. Right? So more than 98% of the oxygen is transported via hemoglobin. And once the oxygen has entered the blood from the lungs, it can be bound by hemoglobin, which is found on the red blood cells. And hemoglobin itself, as you know, is a protein that has four subunits, two alpha subunits and two beta subunits. And each subunit has a heme group in the center. And as many of you know, that iron levels we look at, right, in physiology. So this iron is actually at the center of this heme group, and this thing binds one oxygen molecule. So since we have four, that means each hemoglobin molecule can bind four oxygen molecules. And when hemoglobin is bound to oxygen, it's called oxyhemoglobin. So hemoglobin molecules that have more oxygen typically are brighter red. So that's why if one would compare, you'd see that oxygenated arterial blood tends to be a little brighter red in comparison to deoxygenated venous blood, which is slightly darker. Okay, so this is the hemoglobin molecule with the heme groups which have iron in the center. Okay, so now when oxygen is binding to hemoglobin, there's an interesting phenomenon called cooperativity. So what is that? So when hemoglobin is bound by oxygen, it actually changes its shape. And as more oxygen molecules bind, the affinity for oxygen actually increases. This change in shape that's occurring when initially oxygen starts binding to hemoglobin increases affinity for oxygen. So, so this is known as cooperativity. So when no oxygen is bound, normally hemoglobin is said to be in the tense state or T state. So that is when it has the lowest affinity for oxygen. And then at a point where oxygen starts to bind first, the hemoglobin will have some, conformational changes and it will alter its shape. And this is called the R state or the relaxed state. And this has a higher affinity for oxygen. And because at some point it will cap out, that's why this is a sigmoidal curve. So the percentage of oxygen bound to hemoglobin is related to the partial pressure, which is the PO2. So you have PO2 in a certain, you can have oxygen partial pressures that differ in the lungs versus in the alveoli versus in the blood versus in the tissues, right? And we had learned last time that oxygen in the body moves across a gradient of partial pressures, meaning that oxygen in the gas that is there inside the alveoli, that partial pressure is the highest. Then, as it moves from the alveoli across the cell barrier into the blood in those capillaries in the lungs, and then from there it is transported in the circulatory system to the tissues. Tissues have lower partial pressure relatively, so then oxygen moves through there. And then inside the tissues, it goes to the cells and then it goes to the mitochondria, and the mitochondria also has very lowest partial pressure, allowing this gradient to be sort of uh, existent between the outside to the inside at the smallest level of complexity. So this partial pressure at a given site, and when the oxyhemoglobin, it reaches a tissue that has low PO2, like wherever, like from with respect to alveoli, everything is lower, but imagine skeletal muscle, it will dissociate into oxygen and hemoglobin. And we'll talk a little bit about how this happens. There's an interesting effect that occurs, right? Um, and this results in an increase in local PO2. And then obviously, when it goes the other way around, it reaches tissue that has high PO2. It will continue to take up more oxygen and saturate further and go into further, like because it keeps having higher affinity. And this results in a relative lowered PO2. So that's what you see over here. And there are some factors that affect oxygen affinity. I think we had reached here last time, which is that obviously the pH and PCO2 can affect. So basically, the presence, the PO2 or the pH of the bladder tissue, the relative oxygen concentration. So these things actually affect it. How much hemoglobin is bound by oxygen, how much oxygen it is releasing, and this Bohr effect is explaining that, and I'll go into that in more detail, so I won't talk about it here. But essentially, this ratio and the pH can actually regulate the affinity of hemoglobin for oxygen. So that is the Bohr effect, and there's an alternate effect called the Haldane effect, and we'll talk about that in the next slide or two. Um, so yeah, and then there's this chemical also called 2,3-DPG. It's a chemical found in RBCs and it's a product of the glucose metabolic pathway. And this 2,3-DPG binds to beta chains of the hemoglobin. And this, so then what would happen if you have increased levels of 2,3-DPG is that it would essentially decrease the affinity of hemoglobin for oxygen because it's already bound. And the other way around is that if you have lower levels, it could mean that there's a state of decreased tissue metabolism. So there's fewer 2,3-DPG molecules available to bind hemoglobin. So there's more opportunities for it to bind and therefore have relatively a higher affinity for oxygen. So this essentially follows an inverse relation. And temperature also can affect hemoglobin oxygen affinity. So at increased temperature, for instance, in active muscles like your skeletal muscles, imagine you're running, there's heat generated there. So because of that, it can actually decrease the affinity of hemoglobin for oxygen. So at decreased temperatures, like say a decreased metabolic state, for instance, there's reduced heat production, and so it's the other way around, which means the affinity for of hemoglobin for oxygen increases. And finally, the affinity of hemoglobin also results in a shift in the oxygen hemoglobin dissociation curve, which is over here, right? And an increase in this oxygen affinity results in the curve shifting to the left. And if it's the other way around, where there's a decrease in oxygen affinity, it results in the curve shifting to the right. Okay, so that's shown over here. So you have a higher affinity, there's a left shift. What does that mean? Essentially, that at lower PO2s, you saturate faster, right? And if you have a right shift, meaning there's a reduced affinity, it takes a higher relative PO2 of oxygen for the saturation to reach. And these can be pushed with the factors that we talked about earlier, which is pH and temperature. And another example of something that could essentially tweak this oxygen, the saturation curve essentially is carbon monoxide. So you must have heard of carbon monoxide poisoning, right? People have, I'm sure you've heard of it in the context of someone, maybe facing carbon monoxide poisoning when they were, in a closed, in a closed sort of environment or enclosure of some sort, where they couldn't access enough oxygen and the level of oxygen was decreasing because they were breathing it out, breathing it in, and then, it was not being replaced. So in these cases, it could occur. So this carbon monoxide would also reacts with hemoglobin at the same site which oxygen binds. But the tricky thing here is that hemoglobin actually has a really, really high affinity for carbon monoxide, and it's way, way higher than that for oxygen. So the affinity for oxygen is like so much lower in comparison to the affinity for carbon monoxide. So obviously, if there's a lot of carbon monoxide in some enclosed enclosure where oxygen is being depleted and there's a lot of accumulation of this gas, then it could bind irreversibly to your hemoglobin, and that can lead to all of these symptoms like headache and nausea and tiredness and other things, and eventually someone would like pass out. um, and interestingly, the thing is that respiration rate is usually spared as the partial pressure of oxygen dissolved in the blood, and like CO2 levels increasing is what's detected. But actually, carbon monoxide being bound to hemoglobin won't be immediately detected because we don't have normal sensors in our blood, in our body for carbon monoxide specifically. Um, so another interesting fact here is that hemoglobin, when bound to carbon monoxide, has a cherry red color, and this may be visible in the nails. So the nail bed, which is just like the region, like the skin beneath your nails, and mucous membranes of patients with this poisoning. And so obviously, a person who needs to be treated would be given a lot of oxygen and it should be done as soon as possible, or it could prove to be fatal. So I guess the takeaway here is just to remember how hemoglobin binds to a similar site of oxygen binding on hemoglobin and has a higher affinity for oxygen, which is what makes it so dangerous. And of course, it's irreversibly bound. So that also means that the hemoglobin is just being like consumed when there's carbon monoxide poisoning going on. Right. Okay, so these are just, I think I've continued some of the questions from last time, and I can explain it as we go. We talked about the VQ ratio last time, and these are some of the questions from there. So if you guys remember about the VQ ratio, Do you guys remember? Or do you want me to like explain it again? So, I think you probably like know what it is from last time, but essentially it's a ventilation perfusion ratio, which means it just describes how well like airflow in the lungs matches the blood flow, and it can be at different levels based on the anatomy of the lung. So the apex, the middle, and the lower region of the lung have different values um, of VQ ratio, because essentially it's because of gravity. So you'll have more blood pooling below, so perfusion is better, whereas you have more air above, so you have um, better ventilation in the higher regions. And the middle of the lung is typically where VQ ratio is closest to 1, and on average for an adult, and this is where you guys can try answering what is the normal VQ ratio in a healthy resting adult, and this is the average of the entire lung and not a particular part of the lung. So if anyone doesn't know, what we do is you can either unmute and say the answer, or you can message the answer in the group chat, like in this call, and we'll discuss what the answer is. So does anyone have any idea what the answer might be? Yeah, I'm getting some answers here. Does anyone else want to try? Yeah, so the right answer, yeah, you're right. The answer is C option, which is 0.8. So normally, ventilation is about 4.2 liters per minute. Perfusion is about 5.5 liters per minute. So if you take this ratio, it's for the whole lung on average, it's about zero point 0.8. So this is a really important like clinical diagnostic parameter. Okay, let's try one more relating to this. So in an upright individual, so someone who's standing up straight, um, the pulmonary blood flow will be greatest in which part of the lung? So I just talked about this in relation to VQ ratio. So what do you guys think? Yeah. Yes, you guys are absolutely right. The answer is C, because um, gravity pulls blood downwards. And so if someone is standing, the apex, which means the top part of their lung, would have relatively less perfusion. Perfusion just means how much blood is reaching there. And the base would have more perfusion. So that is what accounts for this regional variation of VQ ratio, right? So yeah, that was about blood flow distribution in the lungs. Maybe we can try another question. So an alveolus receiving blood flow but little ventilation represents what condition? So what's going wrong here? Yeah. Yeah, good. So the answer is B option. Essentially, a ventilation defect means that blood is arriving, but air is not arriving, not in enough quantity. So that would lead to a low VQ ratio. So some examples of like actual conditions where this might occur is like if there's fluid filled inside the alveoli. It could be, someone has some like lower respiratory tract infection or something which causes fluid to be filled inside the alveoli. And so enough air is not reaching, or there could be like an obstruction of some sort that's preventing air from entering adequately into the alveoli. So that causes a ventilation defect. So your V value goes down and your Q, stays the same or compensates for it. And so your ratio overall just becomes lower. Yeah. Okay. Great. So I'm glad all of you are getting it right. Good job. So let's move to the next one. Okay. So oxygenation of hemoglobin in pulmonary capillaries. Um, this is, capillaries in the lung is typically completed within how long? So I think this was talked about in the course material. So the correct answer here is D option, which is 0.01 seconds. So actually hemoglobin becomes saturated like super fast. So it only takes, a hundredth of a second, which is D option, for that to happen. Because if you think about it, right, like if we're breathing and every single time we do so, oxygenation takes half a second, then we're gonna have like a lot of um like we're gonna like the gas is not gonna reach in time. And I think that would not be enough of a safety margin, because RBC transit time itself is like almost a full second. So if RBCs are taking that much time to move by, the saturation needs to happen like super fast. That way there's enough of a safety margin for oxygenation to happen quickly enough that blood keeps moving through, um, and while it's moving by, there is speedy acquisition of oxygen on the hemoglobin. So an RBC will roughly take about 0.75 seconds to move by through by an alveolus. And while that's occurring, you need to be very quick. And zero point 0.5 seconds is not nearly quick enough. So the right answer here is D option. Okay. So, all right. One more question. So I hope that's clear. Yeah. So the next question is about arterial partial pressure. So the slight drop in arterial oxygen partial pressure after leaving the lungs is mainly due to which of these reasons? Right. So, okay. So interesting. So I have some different answers here. So the correct answer for this question is option B. Okay. Which is mixing with bronchial circulation blood. So the reason for this is that some blood will supply the bronchial circulation, so the bronchi, and this does not get oxygenated. Only the blood at the alveoli get oxygenated. So right at the end of this respiratory tree is where the oxygen happens. But this blood that's supplying the bronchi can mix with arterial blood to some degree. This is called a physiological shunt. It's like a normal thing. And in that case, obviously, there'll be a slight drop in arterial PO2 because you have mixing of relatively lower levels of like relatively lower oxygenation in the bronchial blood. So that's why there's a slight drop. Um, yeah, so it's called a physiological shunt. Okay, all right. All right, let's try one more question. So, approximately what percentage of oxygen in the blood is transported bound to hemoglobin? So, I think I already gave this one away earlier when we were talking about hemoglobin. So, I think I won't spend too much time on this one. Yeah, yeah, fantastic. So, all of you got it. Good job. So, the answer is D, which is 97 to 98%. So, almost all, honestly, 97 to 98% is carried by hemoglobin. Only about 2 to 3% is dissolved directly in the plasma. Yeah. So hemoglobin increases like the oxygen carrying capacity by a lot. Okay, I think a few more questions and then we'll talk about some concepts. So the sigmoid shape of the oxygen dissociation curve primarily allows which of these functions? So we looked at the sigmoid shape a little few minutes ago. So what can you really tell from it, essentially? Do others want to give it a shot? Okay, so yeah, the correct answer here, you guys are absolutely right. It's option C, which is efficient loading in the lungs and unloading in the tissues. So this is because of the cooperative binding of oxygen to hemoglobin that when the binding becomes progressively easier, it's what's producing the sigmoid shape. So essentially, as if you remember, in that S-shaped, like right, the S-shaped curve, you have the plateau region. So this plateau region is where there's a high PO2 in the lungs, and so that's like around 100 mmHg. So here the curve is flat. So this means that hemoglobin is almost fully saturated. So that's like 97, 98, almost 100%, right? And even if the lung PO2 drops slightly, the oxygen loading remains high. So this makes sure that there's reliable like oxygen loading in the lungs. And the other region, it's the steep region, right? So it's climbing like this. So that's in the tissues, you have lower PO2. So in the tissues, PO2 is roughly like, I think 40 mmHg. So in comparison to the lungs, it has to be a lot lower, right? So here the curve is very steep. So that means even a small drop in PO2 can cause really large oxygen release from hemoglobin. So this would mean that there's very efficient oxygen unloading, particularly in metabolically active tissues where there's changes in PO2 based on the using up of oxygen. Yeah. So the other obviously options here are wrong because the constant oxygen binding regardless of pressure, it's incorrect because it does bind uh, uh, due to changes with PO2. And then rapid oxygen loss in the lungs, that's kind of the opposite of that, right? So lungs are where there is quite a lot of oxygen, and that's why oxygen loads onto hemoglobin. And finally, the option, I mean, hemoglobin is essential for oxygen transport, so that's not what happened. So yeah, this is option C. I hope it's clear. So we'll move to the next one. Okay, so now if imagine that someone's doing some vigorous exercise, decreased blood pH would cause what kind of effect on hemoglobin? Can you guys tell? Okay, great. So I'm seeing, yeah, you guys are all correct. Good job. So the correct answer is B option, which is that oxygen would be released more readily. So lower pH tends to decrease the affinity. So oxygen would be released more readily. So what happens during exercise is that you have increase in carbon dioxide, which would lead to obviously an increase in H plus ions, leading to a decrease in pH. So it's becoming more acidic, whatever the tissue is, right? Like maybe a skeletal muscle or the blood in that tissue. And so the hemoglobin would be released more readily, and this allows oxygen delivery to be matched to where the demand is in the body for oxygen. So if there's vigorous exercise and the skeletal muscles are uh, relatively more acidic because of an accumulation of CO2, which is leading to an acidification and an increase in H plus ions, the pH decreases and the affinity of hemoglobin and oxygen decreases, and oxygen is released more readily, right? So that's some factors that affect this oxyhemoglobin like curve, right? The saturation curve that we looked at earlier, right? This one, right? So if you have lower pH, you see that there's a right shift. So um, and higher pH, you see a left shift. So that means lower pH means what? Essentially that the affinity is decreasing. So it would take a higher pH to reverse that. Okay, cool. Let's do another question. And then I think we can look at some concepts. So the decrease in hemoglobin affinity for oxygen at a lower pH is known as one of these. So do you guys know what it is? I'll talk about it more. Interesting. Okay, so the correct answer here is C. So this is the Bohr effect. It was discovered by a guy called Christian Bohr. Um, and I will talk about this and the opposite of that sort of, which is the Haldane effect. I'll talk about that as well. So both of them are clear to you, hopefully. Um, yeah, and this Bohr effect essentially just helps active tissues receive more oxygen. Okay, cool. Right. So the Bohr effect, what is it? Right. So as I mentioned, the Bohr effect is describing how there's a decrease in oxygen affinity of hemoglobin in the presence of low pH or high CO2. So those are like one and the same. If you have high CO2, you're going to cause low pH. pH and CO2 essentially therefore have um, a lot of effects on hemoglobin tetramer as we were talking about it, right? So we have at low pH, the histidine residues on one hemoglobin dimer become protonated. So you can see over here, histidine residue, and they become protonated, which permits the formation of a salt bridge between dimers. And this formation of this kind of a salt bridge, you guys would have heard about salt bridges, right, in chemistry. So this stabilizes the deoxygenated T state. So remember we initially talked about the T state and the R state. So the tense state and the relaxed state, right? So this bond that the salt bridge bond, it actually can't form when there is a high pH. So at a high CO2, CO2 binds to these terminal amino groups and forms negatively charged carbamate groups. So this is kind of going into a bit of details, but essentially what happens is these groups stabilize the deoxygenated T state of the hemoglobin by forming some bonds. And so how that uh, in the end it just stabilizes the T state. So it's like um, oxygen is not able to bind because the affinity is decreased, so it's stuck in the T state. It's not able to transition to the R state. And so in that way, essentially by messing with the salt bridge bonds or by forming some other groups that then stabilize the T state in their own way, um, low pH or high CO2 can stabilize deoxyhemoglobin molecule and decrease its affinity for oxygen, which could then lead to the release of oxygen in the peripheral tissue. So this is really important because if the hemoglobin reaches your skeletal muscles, say while you're running, but the affinity for oxygen is still high, what is it going to do? It's not going to be able to release the oxygen. Instead, it might just bind more oxygen because, by cooperativity, we know that if you have already bound oxygen, if hemoglobin is already bound oxygen, you're going to just have higher affinity for oxygen. So it will go to the peripheral tissues and it will just bind more oxygen, and that helps nobody. So having this sensitivity to pH and CO2 is really important for releasing oxygen in the tissues that really need it. Yeah. So that's the Bohr effect. This entire thing, this shebang, is the Bohr effect, the effect of pH and CO2 on oxygen affinity of hemoglobin and how that's important for delivering the correct amount of oxygen to the tissues that need it. Now, the opposite of that sort of is this effect called the Haldane effect. And this is another phenomenon which describes the increased capacity of blood to carry carbon dioxide under conditions of decreased hemoglobin oxygen saturation. So, what does this mean? So, essentially, this Haldane and Bohr effects are the same features of the same phenomenon, but here we're talking about carbon dioxide carrying capacity because you have to remember that hemoglobin does both, right? It carries oxygen to the tissues that require it and carries carbon dioxide as carboxyhemoglobin away from the tissues post respiration in those tissues. Like the CO2 that's accumulating there, it's got to be thrown out. So hemoglobin is carrying it away as well. And so the Haldane effect is what happens to pH and CO2 binding because of oxygen, and the Bohr effect is what happens to oxygen binding because of CO2 and lower pH. So you can see how it's like one is sort of the reverse of the other. So when you have more carbon dioxide binding to hemoglobin at lower oxygen saturations, this would allow the removal of CO2 from the tissue. So what does that mean? In the tissue where you're delivering oxygen, there is lower oxygen saturation. That's what allows the oxygen to be dissociated from there. But this conversely allows the CO2 to be picked up from the tissues, right? So that's clear. And then bound CO2 needs to be released from hemoglobin when it becomes oxygenated. Why? Because the CO2 needs to be released eventually when it reaches near the alveoli, right? So this reverse Haldane effect facilitates elimination of CO2. So it's converse, right? You have lower oxygen saturation, CO2 binding increases. And then the other way around, when you have oxygen saturation, because hemoglobin is acquiring oxygen, that time CO2 is released. So, deoxygenated hemoglobin has a higher affinity for carbon dioxide. Why is this important? Um, because there's an allosteric modulation of carbon dioxide sites, binding sites by the oxygenated heme, and this talks with also the buffering capacity of deoxygenated hemoglobin, which is increased because essentially it ties in with the pH of the RBC cytosol, and then with increased pH, more carbonic acid, so on and so forth. So the total amount of CO2 carried as bicarbonate increases in that case. And compared to if you were carrying carbon dioxide by deoxyhemoglobin, this part of the Haldane effect would actually not play such a major role. So the important takeaway here is essentially that um, at the tissues where you are delivering oxygen, because there is lower PO2, right? Oxygen saturation or oxygen partial pressure. You have oxygen dissociating, and this is regulated by pH and CO2, which is the Bohr effect. And the opposite of that is that there is an increase in binding of carbon dioxide by hemoglobin in these tissues at the same time. So this is what facilitates the transport of oxygen and carbon dioxide from the tissues to the lungs and vice versa, right? Cool. So here's a question relating to these topics that we've covered so far. So imagine that there's someone doing some vigorous exercise, and their skeletal muscles are producing a lot of CO2. So in these, which sequence of events would best explain how this enhances oxygen delivery to the muscles? So you can just sort of apply what we learned so far and let me know what you think. Do others want to give it a shot? Anyone else? Yeah, so you guys are absolutely right. It is option A. So great job. So, which is that increased CO2 lowers the pH, which causes hemoglobin affinity for oxygen to decrease, and so more oxygen is released to the tissues. So this is right, like tying right into the Bohr effect, which describes how carbon dioxide and H plus influence this affinity, right? So during exercise, like this is some vigorous exercise some person is doing, you have a lot of CO2 produced by the muscles and inside the red blood cells. So how does this lead to H plus formation, right? So you guys have probably heard of this reaction where carbon dioxide reacts with water, right? So there's this enzyme called carbonic anhydrase. And during this reaction, between carbon dioxide and water, and the enzyme being carbonic anhydrase, it forms carbonic acid. But this carbonic acid dissociates into H plus ions and bicarbonate. And this increase in H plus, right, is what lowers the pH. It acidifies and lowers the pH. And hemoglobin acts as a buffer and binds some of these H plus ions, but this changes its structure and that reduces the affinity for oxygen. So that's why oxygen is released more readily in these tissues when they need it, when they need it the most. So this is the essential mechanism of the Bohr effect, and I'm glad you guys got it right. So I think this is now clear to everyone. Okay. Okay, let's try one more question. So, so imagine there's a climber, so another person doing some crazy exercise, and this person is at a high altitude and begins to exercise intensely at that altitude. And so the muscle metabolism increases their carbon dioxide production and decreases blood pH. Okay, so that's clear. So, how would the oxygen dissociation curve respond, and what would be the physiological benefit of such a thing? So, give it a think and let me know what you guys think. Yep, good job. So, you guys are killing it. Um, Absolutely right. It's B option, which is that the curve shifts right and it facilitates greater oxygen release to metabolically active tissues. So, what does a rightward shift in the oxygen dissociation curve mean, right? We saw that earlier. I had shown that curve. And if this is the curve and it shifts in this direction, right, that means that that there is a lower affinity for oxygen of hemoglobin, right? And this allows release of oxygen more easily. So again, that is tied in with the Bohr effect here as well. So more oxygen is unloaded from hemoglobin into active tissue in this person. Okay, great. Okay, one more question. And then I think we can move on to the next set of topics. So in systemic tissues, systemic just means in the whole body, carbon dioxide enters RBCs and is rapidly converted into bicarbonate via carbonic anhydrase. So this I mentioned, right? Bicarbonate then leaves the RBC in exchange for chloride ions, and this is called the chloride shift. So how would this process indirectly tie in or contribute to the Bohr effect? So how do these two processes talk to each other? Can you guys think of it? Okay, so I'm really impressed. You guys are absolutely right. It's option C, which is that the generation of H plus ions lowers pH, decreasing hemoglobin's affinity for oxygen. But how does this work, right? Um, so when carbon dioxide is entering the red blood cells, this process takes place, and there's a because bicarbonate is sort of exchanged out for chloride ions, this is preventing bicarbonate buildup. But if bicarbonate is thrown out, the H plus ions still are inside the RBC, right? They're not going out. So they are the ones that bind to hemoglobin, and because of this lowering of pH, that's why this proton binding actually changes the affinity. So if bicarbonate remained, that pH shift would not happen. But because it's taken out, it's thrown out of the cell and exchanged for chloride ions, which would not perceptibly change the pH. That's why the H plus ions are biasing inside the RBCs, and they're what cause the decrease in pH. So this chloride shift is important for acidification within the RBC. Yeah. So maybe I should have written this option a little clearer, which is instead of generation of H plus ions, which is fine. I should have maybe said that they are um, like there's an H plus buildup, and that is more than the bicarbonate. Yeah. So I hope that is clear. Yeah. Okay. So does anyone have any questions about the respiratory system and anything we've covered so far? You can ask me. Um, If not, then I'll move on to the next system that was started in the coursework, which is digestive system. And we'll talk a little bit about the different components of the digestive system, what the cross section of the wall looks like, and about smooth muscles and how they function in the digestive system. So I take it that nobody has any questions. So yeah, all right. Okay. So I'll get started then. So, as you guys must be highly aware, the digestive system is extremely, extremely, extremely essential for us to be able to break down the food we take in and absorb nutrients from the breakdown products. So, there are many different components as you move along what's called the alimentary canal, right? So you start with the mouth, which is important for breaking up your food particles, and you have your salivary glands that moisten and lubricate the food, and you also have enzymes in saliva like amylase that start digesting polysaccharides like starch right away from the mouth itself, even before it's even reached the major digestive organs. Then you have the pharynx, which is important for swallowing the food that is then transported into the esophagus, right? Your food pipe. And that connects via a sphincter over here. And this sphincter is the sort of place where a lot of reflux takes place. Sometimes you feel, right, acid reflux. This sphincter is where things go wrong sometimes. And taking and H. pylori can also cause infections in this region. So you want to take care of your esophageal sphincter. And so then the esophagus leads into the stomach, which is a very muscular bag that churns the food and stores it for some time. While it's doing so, it breaks it down quite a bit. And it also secretes pepsin that digests proteins. And it has acid secretion, as you guys know, I'm sure, which activates the enzymes. It breaks up food, it kills a lot of bugs. And of course, you have goblet cells and stuff that secrete a lot of mucus. So I think there's like peptic cells, chief cells, and so on. I think we'll probably learn about them in more detail in upcoming classes. Um, so yeah, this mucus is what protects the stomach wall from its own acid and makes sure that any bugs that are trying to get through might get stuck in it. Yeah. So there's not a lot of absorption that takes place at the level of the stomach, mostly churning and some amount of digestion detoxification. And then you move to the small intestine from the stomach. The small intestine has three parts, right? The duodenum, which is the first part of the small intestine, the jejunum, and then finally the ileum, which connects to the large intestine, where you have the colon. Um, and the small intestine is what completes the digestion. So it also has mucus that protects it, and it absorbs a lot of nutrients. And we know from the sort of histological studies of the small intestine, we know that there's these villi, right? Those specialized structures which have lacteals and blood capillaries and vessels that allow for absorption of everything from amino acids, fats, sugars, everything, right? But that's after digestion. So you have peptidases that digest proteins, sucrases digest sugars, amylase digest polysaccharides here. You also have lipases, which are coming from the pancreas. So pancreas is a very, very important organ which secretes hormones, or like insulin, for instance, glucagon, right? Somatostatin. These regulate the blood glucose levels. And then the bicarbonates from here neutralize the stomach acid, which is important because here it's like really, really acidic. And then it also secretes amylase and lipase. And the liver is very important. We all know it. The liver detoxifies. We've heard about this so many times. And it's indeed the case. It destroys old cells, it destroys poisons, it metabolizes a lot of drugs, like medicines that you take go to the liver, which is why people sometimes tend to say that, okay, don't take too many medications. The best thing to do is to limit it because the liver is under a burden. Of course, this is not always the case. If you're in pain, just take a crocin, but like it's it depends. Maybe also on the age of a person, on their already on the health of their liver already. um and like if a person drinks a lot, they could have liver cirrhosis, and so they already have a lot of pressure on their liver. So in these cases, obviously, you have to think differently. And obviously it secretes bile, which is a very important mix of uh, 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 um, compounds that aids in digestion. And this little guy here is the gallbladder, which stores and concentrates the bile. Though people can obviously live without it because people get gallbladder stones and then it's removed and then they're still just fine. So it's important, but I mean, it's okay. Yeah. So yeah, and then eventually in the intestine, once most of the digestion and absorption is done, there's some reabsorption of water, it forms feces, and then it's thrown out of the body. So that's your digestive system and all its parts. And now lining these um, walls of the alimentary canal are a lot of different components. One of them is smooth muscles, which we'll talk about in some detail. Um, and they are what facilitate the peristalsis, which is moving along of the food while it's being digested through the system. Peristalsis is like sort of like an alternate undulating muscular contraction event. It's not under our voluntary control, it's taking place on its own, based on involuntary nervous signals, and we'll learn about those. So the digestive tract, if you were to, take a section of it like this, you would see that there are actually several different layers. So here in the inside, you have the mucosa, the inner side, you have the mucosa consisting of the mucous epithelium over here, the lamina propria, and then you have this muscularis mucosae layer. After the mucosa, you have the submucosa, where you will find ducts from glands, you'll find, and the gland itself will be there in the submucosa. And then you move to the muscularis layer, which is basically the muscle layer. So you will have circular muscles and longitudinal muscles. that you'll find in the muscle layer, and we'll talk about, what sort of type of muscles these are. They're smooth muscles, right? So we'll learn about them. And finally, the outermost one, you have the serosa, which is the connective tissue layer, and it forms like it's connected with the peritoneum, and this is like the outer protective covering. And you have nerves innervating this tissue, which is where the impulse for peristalsis and other contraction relaxation events take place, and blood vessels, and this is the layer of the mesentery. You also have lymph nodes, and in some places you have interesting secondary lymphoid structures like um, mucosa associated lymphoid tissue, which you guys might have heard of, and I'll cover in the extra class that I'm taking on the immune system. Okay, so a few questions about what we've seen so far, and then we can proceed with learning about the smooth muscles. Okay, so the vertebrate body plan is often described as a tube within a tube, right? So, which of the statements that I'll provide explains what the functional significance of such an organization in the digestive system entails? So, have a think and let me know. Oh, I had a question here. Oh my goodness, I missed it. I'm so sorry. Um, can you explain about the lung microbiome, like in asthma? Uh, you know what? Yeah, I'll explain right after this question. My bad if I missed the question somehow. Lung microbiome. Interesting. Asthma. Hmm. So, asthma is not necessarily caused by. Okay, so for anyone who has not seen it, like Aditya has a question about lung microbiome, um, and whether it's like linked to asthma. I believe that's your question. So, asthma actually has many causes. It doesn't necessarily only relate to microbiota of the lungs. It could be environmental triggers that cause, some sort of um, spasming of the upper respiratory tract, which can lead to reduced air intake, and that's what causes that heavy breathing and the and the wheezing and the hyperventilation, which makes you need to take salbutamol. But um, it could be environmental triggers, it like dust or animal dander or um, you know, spores from like trees or fungi, or even it could be some food. Some people get allergies because of food, which can cause asthma-like symptoms. It could even be because of altitude. It could be because the air is too dry. That happens to some people. I had a friend who it used to happen to like that. Like if she went into a really dry air, that would trigger asthma. And then you'd have to take like inhaler for that. Um, but microbiota specifically, I don't know whether the lung microbiome can affect asthma or not. Maybe I need to look into the literature myself because I don't really know whether lung microbiome can affect um, asthma. I can look it up and let you know in the next class if you like. I guess if you have like an infection, um, like in the lung, which changes the microbiome, it could trigger some asthma like or even the development of asthma. I don't see why that's not possible, like because even small changes in the gut can trigger, problems with digestion, and it can and trigger all sorts of issues. So I think if you did have a change in sort of the microbial community that's present in your lungs with respect to whatever bacteria or virus or fungi, that could, I suppose, be linked to asthma in some cases. But yeah, I would need to look into that in more detail. I'm not sure. I don't know what the like clinical sort of consensus on it is right now. Yeah. Okay. Anyway, I hope that is. Oh, hold on. I just thought of something. So okay, so if you have some sort of allergic reaction, which could be because of lung microbiome changes, because there'd be some sort of immune system sort of dysregulation or modulation, that could definitely cause, so allergic inflammation because of some infection or something, that could definitely cause asthma-like symptoms. So yeah, there is that. And I think there is this hygiene hypothesis, right? That people who are sort of conditioned, maybe in like suppose developing countries, people who are conditioned to a lot of different types of um, like microbial like organisms and um, environmental allergens from a young age, they have a lower chance of developing asthma, whereas people who grow up in, say, a higher a cleaner environment for all intents and purposes, maybe in more developed countries that has become the case, then they actually have a higher asthma risk. So childhood sort of exposure to things can actually affect how likely you are to develop some problem or disease. And in some cases, it has been linked that if you are exposed to more of these pathogens at a young age, obviously not like an infant stage when you don't have an immune system, but like at a younger age, that could keep you more immune later on. Yeah, cool. So I hope that is not too disappointing a answer for you. I can look into it more, but this is all I know for now. Okay, cool. So let's move on to the next question. So we were just talking about the layers that we saw in the alimentary canal, right? So we looked at that diagram. So what is the order of layers of the alimentary canal from when you're moving from the outside, so the outermost layer, to the inside? So what do you guys think? Okay, so good job. You guys are right. The answer is A option, which is if you move from outside to inside, the correct order of the layers is first you have the serosa. So we can just look at it here, right? First you have the serosa, then you have these muscle layer where you have the longitudinal muscles, and then you have your circular muscles here. So the longitudinal muscles, then the circular muscles, right? And then you have the submucosa, which is this guy over here, and then you have the mucosa. So that is absolutely right. Good job. So I hope now you remember this. Let's look at another question. So I have not talked about this, so I'll talk about it once we complete this question. So there's this thing called the myenteric plexus, and it regulates certain functions. So, which ones does it primarily regulate? Yeah, so that's great. You guys are right. It is C option. So the myenteric plexus or the Auerbach's plexus, it's also called Auerbach. I might be pronouncing it wrong. Um, it lies between those two layers in the musculature, which is the longitudinal and the circular muscle layers, right, that we saw in the gut wall. And its main function is to coordinate the smooth muscle contractions that are important for peristalsis. So the peristalsis are these like rhythmic waves, right, that push the food along the digestive tract. So there's something else which is called the submucosal plexus, and that mainly regulates like secretions and local blood flow. So the myenteric plexus is like it's kind of like a hub between the two muscular layers that's important for coordinating peristalsis. Okay, cool. And I think it was there in that diagram, isn't it? Yeah, there you go. So this is the myenteric plexus and this is the submucosal plexus. So this guy is important for secretions because it's found where a lot of these ducts and glands and stuff are there. And then the myenteric plexus, which is in the next layer, in the muscularis layer, that's important for coordinating um, peristalsis. And both of these are grouped together as intramural plexus. Okay. Okay, let's try another question, sort of relating to innervation. So the activation of the parasympathetic nervous system, right, in the GIT typically results in which of these events? Yep, I think you guys got it like right away, which is great. So you're right. The correct option is option B, which is increased peristalsis and increased digestive secretions. So, what is happening here is that if there is an activation of the parasympathetic nervous system, and this happens particularly through this nerve called the vagus nerve, right? It generally stimulates digestive activity. It increases peristalsis, secretion of digestive juices by releasing acetylcholine, right? So it releases acetylcholine onto enteric neurons and onto the smooth muscles, and this is what promotes digestion, nutrient absorption, and this is very important for this role that you it's like rest and digest role, right? So someone is resting, they're they're digesting because their parasympathetic nervous system is active, and that is it's not completely accurate to say this, but in general that is sort of what's going on, which is that if you're resting, the parasympathetic the sort of the blood and the nervous system is redirecting the efforts to digesting food, right? Like if you're running, that's not the case. If you're running, your body doesn't care that you need to digest food. Your body will send blood and nervous impulses to your skeletal muscles and, to your respiratory system to make sure that you don't die. So, yeah, so rest and digest is like a phrase that you can use to remember essentially that when you're resting, your parasympathetic nervous system is active, and that is then talking to the GIT to increase peristalsis and increase secretion of digestive secretions that helps you digest food. Good. All right. Another question about the nervous system here. It's about sympathetic stimulation. How does sympathetic stimulation in this turn generally affect the GIT? So, what's going on here? So, this should be relatively easy to put together. Yeah. So, this one is, it sort of follows from the previous question. It's the opposite of rest and digest. So, if you have sympathetic stimulation, you are inhibiting intestinal motility and reducing secretion. So, the correct answer here is option B. So, the sympathetic nervous system generally would inhibit digestive activity. And how does it do so? So, if you have, the postganglionic sympathetic neurons, they release norepinephrine, and this then reduces intestinal motility, it decreases the secretions, and it constricts the blood vessels that supply the gut. Just remember that when you're running, say you're running away from a dog or you're just running in a race, the thing your body cares least about is digesting your food. So that's why the blood vessels are constricted near the gut, so your blood is not being redirected to your gut anymore. And we learned about this when we talked about the circulatory system, right? Where the body can very cleverly decide where the blood is needed the most. And we have all the systems working together on that, right? The respiratory system, you have all these Bohr effects and all at the level of the blood. You have neural effects that then constrict. So you have muscular and neuromuscular effects, and the it's crazy. This everything works together to make sure that the right tissues are getting oxygen or blood at the right time based on what is the need of the body. Everything is coordinated. It's crazy. Yeah, so this system essentially allows the body to redirect your energy and your blood flow towards the skeletal muscles during like maybe some stress or like a fight or flight response, right? So the correct answer here is option B. Okay. All right, great. So we talked about that. Now we can go a little bit into the smooth muscles. So we had learned earlier when we were talking about muscles, we learned a lot about skeletal muscles and how they look. We talked about cardiac muscles and how they're slightly different from skeletal muscles, but overall have a lot of similarities in their anatomy and morphology. Smooth muscles actually look very different, and they also have, and this impacts how different they are functionally as well, right? So smooth muscles, why are they called that? Because they don't have any visible striations, right? The striations that were caused by the isotropic and anisotropic bands that we saw in the skeletal and cardiac muscles, those aren't there here. So that's why they're called smooth muscles. And they're present in a lot of these hollow organs. They're found in blood vessels, they're found in the eye, they're found in the skin, and of course, they're found in the digestive tract, the alimentary canal, which we will learn a little bit more about. So these smooth muscles, as we know, display involuntary control. So we cannot actively make our smooth muscles contract because we want them to, right? It's going on based on triggers from hormones, neural stimulation from the autonomic nervous system, and different local factors, right? So in certain locations, like in the walls of visceral organs, like in the organs of your digestive tract, um, the stretching of the muscle also can trigger its contraction. And in general, if you see, this is a histology slide and this is a diagram. So this is a comparison, right? So this is the muscle in your stomach. And this is, if you took a little section of it, this is what it would look like. And this is how different it looks in like a joint. So smooth muscle fibers are themselves spindle shaped. You guys must have seen in school, right? So the cardiac muscles and the voluntary muscles have like a sort of band like look to them, but these guys are spindle shaped and they are not multinucleate. So they're not sort of fused all together, having one big sort of um, like one big sarcoplasm mixed and then many, many, many nuclei. These guys have single nuclei, a single nucleus per cell, and they can be about 30 to 200 micron in size. And they form these sheets of tissues. They're very coordinated and they are sort of held together by a lot of gap junctions between the cells. So, gap junctions essentially allow for a sort of nice functional connection between smooth muscle cells in the tissue. Right. So you can have two different types of organizations of smooth muscles. You can have single unit smooth muscles or multi-unit smooth muscles. So what does that really mean? So unitary smooth muscles or visceral smooth muscles or single unit smooth muscles, this is typically observed in the walls of hollow organs, and they are important for like slow steady contractions that like for instance like peristalsis that allows substances like food in the digestive tract to move through the body. Meanwhile, multi-unit smooth muscles, this is like the second type of smooth muscles that we see in the body. They are composed of cells that rarely have gap junctions, meaning functional connections between these cells are rare. So they're not electrically coupled, right? Because gap junctions, they exchange a lot of electrical and even chemical signals. And in this case, since there's not much electrical coupling, they are not electrical because they don't have gap junctions, right? So because of that, contraction doesn't spread from one cell to the next like it would in single unit ones, but it's confined to that cell that was originally stimulated. So it's like each cell is a unit, whereas here each of the cells come together to form a single unit. So that's how it works, right? So it is a little bit counterintuitive sounding, but this is how it works. Okay. So this type of smooth muscle is observed in the large airways to the lungs, in large arteries, arrector pili muscles that are there with hair follicles. These are the guys that are important for, when suppose you feel cold and your hair stands, right? Or you're scared and your hair stands, or, like there's whatever some or you're angry and your hair stands or something like that, right? So in these regions, that's this muscle, arrector pili muscle, which is connected to your hair follicles, is what actually makes you get like goose skin or whatever, like get yeah. And it's also there in internal eye muscles, which is very important for regulating light entry into your eye and the shape of the lens and so on. So these are where each muscle has like a connection, whereas these guys have these um, have these gap junctions and these varicosities that help to transmit electrical information across them. So these are all working together, whereas these guys are working a little more independently. Okay. So yeah, that's about single unit and multi-unit smooth muscles. Yeah. So a little bit more about what you would see if you looked at smooth muscles. And how they look like in the relaxed versus contracted state. So why do they not have Z discs and how do they compensate for the absence of Z discs, right? So Z disc again, just as a reminder, in skeletal muscles and cardiac muscles, Z discs essentially are the They're like the unit of the skeletal muscle, and they are the contractile unit, and they're composed of A and I bands and a bunch of other things, H line, M line, and so on. Okay, so these guys don't have striations, but they still have actin and myosin contractile proteins, because how would they contract otherwise? And these interact to generate tension, but the fibers are not arranged in an orderly fashion like they were in sarcomeres. And so because there's no striations, but instead they are anchored to these things called dense bodies. So you can see the dense body here, that dot over there. So this is, which are scattered sort of all around, um, in the sarcolemma, sorry, in the sarcoplasm, and anchored to the sarcolemma, which is the cell membrane of the cell. And there's this network of intermediate fibers. So intermediate fibers, so you guys have heard about actin, right? And then you guys have heard about microtubules, probably in context. So actin and microtubules give shape to the cell. They help during division. Microtubules have the spindle fibers. Actin is important for migration and contraction, with the help of actomyosin contractile filaments, right? Intermediate fibers is another element of the cytoskeleton, and they're literally that. They're intermediate in terms of length, in terms of tension, right? And these guys are important because in this case, because they run between these dense bodies, and they provide like an internal framework. So internal intermediate fibers are interesting. So if you I have imaged them before, and they look like a cage, I had looked at the protein vimentin, which is an important sort of component of intermediate fibers. And if you look at it in cells, they look like cages, because they're literally providing a framework within cells. So if you look up something like vimentin cage on Google, if you're interested, they're really beautiful, and they actually look like cages, because they're providing like a structural framework. Um, yeah, so this is what these guys are doing. They're sort of forming a network between these hubs, which are the dense bodies, and this is the framework for contractile proteins to work against. So this dense body is analogous to the Z disc of skeletal muscles, and it's what anchors the thin filaments in position. And in this case, calcium ions are supplied primarily from the extracellular environment. So, not from, like, in skeletal muscles, we had the sarcoplasmic reticulum providing a majority of calcium ions. It was different for cardiac muscles, if you remember, right? Based on how developed the sarcoplasmic reticulum is and how large the D tubules are. In this case, calcium ions mostly come in from the extracellular environment, and T tubules are absent, but some small indentations called calveoli, these are there in the sarcolemma, and they are locations where a huge amount of calcium channels are present at high density, and those are the guys that facilitate calcium entry into the cell. And the SR, the sarcoplasmic reticulum, is there, but it's not as developed. So it sort of looks like, the skeletal muscles or striated muscles are on one side, the smooth muscles are on the other side, with these differences, and then the cardiac muscles are kind of like best of both worlds of both of them, right? Which makes some sort of, philosophical sense, considering you'd need whatever is best for the heart. And finally, an interesting, sort of difference at the molecular level, which is smooth muscles, they don't have troponin. So, um, but cross bridge formation still happens. So while the troponin tropomyosin complex, which was very important for displacing calcium, if you guys remember, right? That doesn't happen in the same mechanism like it did in skeletal muscles. Here, instead of troponin and tropomyosin complex doing the job, you have a very important regulatory protein called calmodulin. So what happens here is that when a smooth muscle cell is stimulated, there's, as we talked about it, a lot of external calcium ion moves through those calcium channels, in the sarcolemma, and a lot of additional calcium is released from whatever little bit of the SR is available. And this calcium will bind to calmodulin in the cytoplasm, and it'll form this like complex. And this activates an enzyme, which is MLC, MLC kinase. So myosin has like light chain and heavy chain. So myosin light chain kinase is activated, and when this is activated, it activates myosin heads. How does it do that? Kinases do what, right? It adds phosphate groups to stuff. So it converts ATP to ADP, and this P that is released, that's the thing that they use to phosphorylate substrates. So when this MLCK is activated, it converts ATP to ADP, and a phosphate group is released, which is then used to phosphorylate the myosin head. And that's what activates it. So once that's done, the head can then attach to the actin binding sites and pull on it. Yeah, and these thin filaments, when they're pulled, they slide past the thick filaments, and then they pull on the dense bodies, which then pull on the intermediate network, and then that is propagated throughout the sarcoplasm of that cell. And this arrangement allows the entire thing to sort of contract, but it's sort instead of sort of pulling in the way that we were accustomed to seeing in skeletal muscles, they sort of just kind of twist into itself. So it sort of causes this bulge and this corkscrew like turning. And so this is what it looks like. So you have this relaxed muscle cell, and then you have calcium coming in, binding calmodulin, activating MLCK, phosphorylating myosin head attaches to actin, pulls on the thin filaments, they pull dense bodies, um, which then pulls this entire network of intermediate filaments. You can understand how that's happening, right? And this sort of twists the cell and makes it contracted, and then it looks like this weird little corkscrew, which is bulged in the middle. So that's how the smooth muscle contracts. And then obviously, if it is a single unit, then this would have propagated to many other cells. So they'd all contract at the same time and look like this. And multi-unit would be sort of more individual. Right. So with that, let's start some questions um, relating to what we have seen so far. Okay. So which of the following features is a characteristic of smooth muscle cells? Yeah, I think I made this one kind of obvious. um, yeah, so the correct answer here is B option. Uh, Smooth muscle cells are spindle-shaped cells containing a centrally located nucleus. Yeah, so they're tapering at both ends, and they don't have striations. They're not organized into sarcomeres. And yeah, that's why, sort of these slow, sustained contractions are um, possible in these kind of cells rather than, the rapid, sort of forceful contractions that you see in skeletal muscles. Okay. So we talked about the gap junctions, right, in spoon muscle. So what is their primary purpose? Anyone? Yeah, good job. So you guys are absolutely right. The answer is option A, which is that they allow electrical signals and ions to spread between adjacent cells. Um, yeah, and these are, particularly important in the case of single unit smooth muscle cells, right? Because that's what allows them to contract sort of more homogeneously. Okay, let's try one more question. So, which of the following tissues is most likely to contain multi unit smooth muscles where each cell, as I mentioned, receives independent neural inputs? So, I talked about some of them. So, do you guys remember? Yeah, great. So, I think all of you um, have absorbed it really well. It is option C, which is the iris of the eye. So, these multi-unit smooth muscles, because it's like individual cells, these are the ones that are found in tissues that require a really like precise control of contraction. So in these tissues, like the smooth muscle cell would receive its own neural input rather than sort of rely on coupling at the level of the gap junction. And so that's why a classic example is some tissue that requires really precise modulation, because modulating the amount of light entering the eye is extremely, extremely important. And so it's found in the iris of the eye because that can finely control the pupil diameter um, at all times. Yeah. All right. Let's look at another question. So this is like a bit of a thinking one. So if a researcher, imagine, right, records the membrane potential of a smooth muscle cell and observes that there are these rhythmic oscillations between minus 50 millivolts and minus 40 millivolts, even without any stimulation, right? Which interpretation that I've listed below would best explain this observation? Okay, great. Yeah. So, glad to see you guys have got it right. It is B option, which is that the smooth muscle cells exhibit like spontaneous slow waves that can trigger contraction when stimulated. So smooth muscle cells have this thing where they have spontaneous fluctuations in membrane potential, and these are called slow waves. Okay, so these oscillations occur like even without any stimulation, and they represent like these periodic changes that occur in like ionic uh, ionic permeability. So while slow waves themselves may not trigger contraction, a stimulus like stretch or neurotransmitter release can amplify them and produce like proper action potentials that can lead to contraction. Wait, I think someone's trying to like join. Someone has a question. Explain elaborately. Okay, just 1 second. Yeah. So basically, there's something called basic electrical rhythm, which is also like another way of describing these slow waves. So they are these fluctuations that are quite rhythmic actually, that occur in the membrane potential of the smooth muscle cells, and they don't directly cause contraction, but they sort of set a baseline of excitability, and like specification of timing for the contraction. So they are just I wish I had an image here to show you guys. Maybe I'll show it next time to explain better. But there's these like if membrane potential is like, imagine this is the graph and there is some threshold potential, right? So the membrane potential will take you over the threshold, which actually generates an action potential, right? But in normal, when there's no act in the absence of any stimulation or action potential, there is still some oscillation that's below this threshold. Okay. And that helps with setting the timing, because if, say, there is a slight up in the oscillation, this up, if you further stimulate it, it will immediately go above the threshold potential. So it's setting like this rhythmicity is allowing you to set a timing and an excitability for the contractions that would require more further stimulation. So there's this cycles of repeated depolarization and repolarization. So typically what is happening is at the resting state, you have membrane potential at around -50 to -60 millivolts. And if you have the slow wave depolarization, that can oscillate from -50 to -40. And then if you have a spike potential, that will just spike it further -40 millivolts over the threshold. So they alone do not cause contraction, but if there is a spike potential on top of the slow wave, that immediately throws it um, over the threshold and allows the action potential to then cause contraction. Yeah. Okay. So that's that. All right. Let's move to one more question. I think we'll do like one or two more questions and then sort of adjourn for today. Okay. So, which of the following statements about smooth muscles and skeletal muscles is not a difference between the two of them? So, can you guys figure out based on what we discussed earlier? I hope my previous explanation was fine, yeah? Like, if you want me to explain more, you can let me know for the previous question. Yeah, so great. The correct answer here is A option, which is that they both rely on actomyosin interactions or for contraction. So why are the others wrong? Skeletal muscles contain sarcomeres, whereas smooth muscles do not. Um, so this is correct, right? And then smooth muscle contraction involves calmodulin, whereas skeletal muscles require tropon and tropomyosin. And finally, skeletal muscle contractions are generally faster, and smooth muscle contractions are slow and rhythmic, right? Explain. I didn't understand. Okay, question number 30. Okay. I'll tell you what. Let's discuss like another question and then I'll come back to it. But basically, okay, so actually, um, so question 30 is relating to, so if you maybe read up about it, essentially it is about a phenomenon called slow waves. So there is a baseline sort of oscillation in in repolarization and depolarization that is seen in these smooth muscles. Okay, this is different from skeletal muscles. In smooth muscles, it's there. So there's this like rhythmic, like you know how if there's a enzyme kinetics reaction, there is some certain threshold of energy that you need to cross for the reaction to take place. Correct. So here also there is a threshold above which you will only have an action potential. So what happens in smooth muscles is that there is this below threshold level of oscillation where ions are fluxing in and out, but it's and it's called a slow wave. So full-fledged contraction is not happening, but it's so close to the threshold that if there is this thing called a spike potential, where essentially you'll just have a spike of ionic entry, you will have contraction. Okay. So this is just an interesting characteristic of smooth muscles that helps maintain the rhythmicity of slow contractions. Um, yeah, because there is an oscillation in ion conductance, so there's an oscillation in this low level depolarization and repolarization. So there is some like slow wave frequency in different regions of the GI tract. Like in the stomach, you have every 3 minutes or so. In the duodenum, you have every 12 minutes or so. And I don't know for the rest of the parts what it is. So this frequency is basically sort of decreasing as we move along from the stomach to the intestine. So slower contractions are encouraged because it's like it's like determining a timing, because say like imagine a sine wave, right? In the sine wave, you have definite intervals between your maximas, right? So, and imagine that now to the sine wave, you're giving a little kick at every maxima. So the sine wave, the low level sine wave is already determined where the kick will be. The kick itself is then coming and pushing it over the threshold, right? So yeah, that's that. I hope that's clear. If not, then I'll explain in next class. Okay. Um, all right. Let me see. How many questions do I have left? Okay, so we quite a lot to cover. So maybe for today I'll wrap it up, and then we can look at more questions in next class because we've been going for quite some time now, right? So, all right. Uh, Great. Maybe we can finish these questions. What do you guys think? Yeah, let's just do a few more. Just three more questions and then we'll be done for today. Okay, so in smooth muscle cells, what is the primary function of dense bodies? It's an easy one, so we can do it. Anyone? Uh-huh. Great. Good, good. Good job. So it is B option. The dense bodies function to anchor the actin filaments and transmit contractile force. So they sort of are like analogs of Z discs that you see in the skeletal muscle. Great. Okay, one more question. So compared to the skeletal muscles, Smooth muscles can shorten quite a lot. So why is that? What could be one reason? Yeah. Yeah, this one is pretty easy. So it is indeed option C. The actin filaments are arranged in multiple orientations throughout the cell, and not rather than, in those sort of parallel arrays that we were seeing in skeletal muscles. And so the myosin can pull actin filaments in all different directions, allowing the contraction to happen, and it sort of shortens very dramatically. Uh, so the cell can actually go to about, like, I think, one third of its original length. So that's how much sort of it can get squeezed inside. Not squeezed, but pulled, and become smaller. Okay, one last question before we wrap up. So the smooth muscle contractions, so just look at this statement. The smooth muscle contractions are generally slower than skeletal muscle contractions, but they can be sustained for longer periods using relatively little ATP. So, does this statement sound true or false to you guys? And yeah, yeah, yes. Thank you for the enthusiastic yes. It is true. Um, yeah, good job. So, this is a true statement, and that's what we've been talking about so far. Which is that the smooth muscle contractions are slow, like we talked about the slow waves, right? But they are, and during that time they're highly energy efficient, so they can keep, contracting and relaxing for long periods, and they consume very, very little ATP. Because of this mechanism called the latch state, which allows actin and myosin to sort of remain attached for a long period of time without ATP hydrolysis. We'll talk about the latch state in more detail in the next class. Yeah. So, great. That's, I think that's all for today. We can, yeah, we can wrap it up.
Thank you.
Yeah. Yeah, no problem. If anyone has any questions, please let me know, and I'll address them in the next class. I think we'll continue with this, and then there'll be stuff about, digestion, like in terms of enzymes and all. All right. Okay. I'll see you guys next class then. Have a good week. Bye bye.